Pharmapal.
Ligand-protein cross-docking with water molecules. Hello everyone! Welcome back to our channel. Today, we're diving into an exciting topic in the world of molecular biology and drug design, ligand-protein docking and the crucial role of water molecules in this process. We'll be summarizing key findings from a fascinating paper by Ramasamy Thilagavathai and Ricardo L. Mancera, published in the Journal of Chemical Information and Modeling. First, let's break down what we mean by ligand-protein docking. In simple terms, this is a computational method used to predict how small molecules, called ligands, bind to a receptor of known 3D structure, typically a protein. This process is essential in drug discovery, as it helps scientists understand how potential drugs interact with their target proteins. Now, you might wonder, why are water molecules important in this context? Well, it turns out that water isn't just a passive player in biological systems. The paper highlights that water molecules can significantly influence the binding accuracy of ligands to proteins. Water molecules as mediators, they can form hydrogen bonds between ligands and proteins, stabilizing the binding complex. Conservation across structures, many water molecules occupy conserved positions in protein structures, which means they can be crucial for binding interactions. Let's delve into some of the key findings. Cross-docking simulations. The authors performed simulations using various ligand-protein complexes, focusing on those with water molecules and their binding sites. Inclusion of water molecules. They found that including common water molecules in docking simulations significantly improved the accuracy of predicting how ligands bind to proteins. Statistical significance. The results showed a statistically significant increase in docking accuracy when water molecules were considered, underscoring their importance in molecular docking studies. So, how did they conduct their research? Here's a brief overview of their methodology. Selection of complexes. They selected ligand-protein complexes from the protein data bank, focusing on those known to benefit from water inclusion. Common water molecules. By analyzing crystal structures, they identified common water molecules that appeared in multiple complexes. Docking simulations. They conducted cross-docking simulations, comparing results with and without these water molecules to assess their impact on binding accuracy. The study revealed that increased success rates. The inclusion of water molecules led to a higher success rate in accurately docking ligands, with improvements in the root mean square deviation, RMSD, values, a measure of prediction accuracy. Practical applications. These findings have practical implications for drug design, suggesting that computational models should incorporate water molecules to enhance the reliability of docking predictions. Next, we're diving deep into the fascinating world of molecular docking, specifically focusing on the figures and tables from a pivotal paper on ligand-protein interactions. In this study, the authors investigate how including water molecules in docking simulations can significantly improve the accuracy of ligand-protein interactions. They conducted cross-docking simulations on various proteins, analyzing the impact of water on binding accuracy. Key terms. Molecular docking, a method used to predict the preferred orientation of one molecule to a second when bound to each other. Cross-docking, docking multiple ligands from different crystal structures onto a single protein conformation. Figure 1 docking results for lysozyme. Let's take a look at Figure 1. Here, we see the docking results for the lysozyme ligand complex. Visual description. The native binding mode of the ligand is shown in different colors. Green for the top-ranked pose with water molecules included. Red for the pose without water. Cyan represents the water molecules themselves. This figure illustrates how the presence of water can alter the binding pose of the ligand, indicating a more favorable interaction when water is included. Key insight. The top-ranked binding pose with water shows a root mean square deviation, RMSD, improvement, highlighting the importance of water in enhancing docking predictions. Next, let's discuss Table 1, which summarizes the docking simulations for various ligand-protein complexes. Analysis. The table clearly shows that in most cases, the inclusion of water molecules leads to a lower RMSD, suggesting a more accurate docking prediction. Key takeaway. This data reinforces the author's conclusion that water molecules play a crucial role in ligand binding and should be considered in docking simulations. Now, let's move on to Figure 2, which depicts the accuracy of cross-docking simulations. Visual elements. This figure uses bar graphs to compare the success rates of docking with and without water molecules across different proteins. Observations. Proteins like D-xylosisomerase show a marked increase in success rate when water is included, emphasizing the necessity of considering hydration in docking studies. To wrap up, the findings from this research highlight the significance of incorporating water molecules in ligand-protein docking simulations. The results suggest that doing so significantly enhances the accuracy of predictions, which is crucial for drug design and discovery. Thank you for joining us today. If you found this video informative, please give it a thumbs up, subscribe for more content, and let us know your thoughts in the comments below. Until next time, keep exploring the wonders of science.
Pharmapal.